नमस्कार। आप सभी लोग अपने अपने घरों पर, अपने अपने स्थानों पर कोविड-19 से बचने के उपायों के साथ साथ सकुशल होंगे, इसी कामना के साथ हम वापस अपने टॉपिक पे चलते हैं। एज स्टूडेंट वी हैव टॉक अबाउट इन आवर प्रीवियस लेक्चर about the paleotropy and the polygenic traits, and I have also told you that you must try to make the cross for the mulatto, right? That is, mulatto is most important cross in your point of view because it can show the 29 distinct genotypes, as well as in the previous one in diploid cross you have done only, that is the 9 genotype one, right? And after the rediscovery of Mendel's law, on the basis of the discovery, right, we have to just come on the chromosomes structures. Means when the hereditaries and currents and one stomach can show the Mendel's law, right? But after that, the discovery of the chromosomes, there is another theory can be proposed, that is called the chromosomal theory of the inheritance. This chromosomal theory of inheritance, we can say, that can be always proposed by independently by the scientists Walter Sutton and the Theodore Boveri. Means they can be capable to, they provided this chromosomal theory. We can say they united, means their united knowledge, right, of chromosomal segregations with Mendelian principles, right? Means they can be just united, they can be combined, the chromosomal segregation with the Mendelian principles, and that is called the chromosomal theory of the inheritance, right? So we all must know the, they have some important points, most important points which can be asked so many times in examinations. We can say the main points of this theory. The first of all, we can say the gametes carry and transmit hereditary character from one generation to another generation. Means he given that the gametes can transfer, they can carry, the transmit hereditary characters. Means gametes transmit the hereditary characters from one generation to the another generation, right? And the second, we can say the nucleus is the site where the hereditary factors are present. Means generally inside the cell of nucleus, right, where the different factors, hereditary factors are present in there. And we can say after that, on the third basis, third point, the chromosomes as well as the genes are found in the pairs. Means generally according to the chromosomal theory, he also proposed that the chromosomes are as well as the genes are always found into the pairs. And later on he also explained that the two alleles of a gene pair are located on the homologous site on the homologous chromosome. Means two alleles, listen carefully, that two alleles of a gene pair, दो एलिलीज जो होता है किसी भी जीन पेयर के, दे कैन बी लोकेटेड ऑन द होमोलोगस साइड, एक ही साइड पे होते हैं ऑन द होमोलोगस क्रोमोसोम्स during the cell division, during the, we can say, meiotic cell division or stages of the anaphase first. Means the anaphase first, the separation of homologous chromosome take place. Means generally while the anaphase, during the anaphase first meiotic stage, when the separation of homologous chromosome take place, in that particular time the two alleles of the gene pairs are located on the homologous site on the homologous chromosome, as well as on the next point, that the sperm and the egg having haploid set of the chromosome fuse to regain the diploid state. Means during when the sperm and egg, generally they contain the haploid sets of the chromosomes, and after the fusion they can going to become the diploid stage in their structures on the basis of chromosomal theory. And later on we can say that the homologous chromosomes synapse during the meiosis and get separated to pass into the different cells, right? When the homologous chromosome can be attached to each other, right, during the meiosis conditions, they can be get separated, right, and pass into different cells, and they can be, after separation they can pass on different cells. right? It is generally on the basis of the segregation or independent assortment. As I have told you, after the crossing over, after the mixing of these genes, they can be separated out from each other, right, in the meiosis conditions. And so the Sutton and Boveri said that the pairing and the separation of pairs of the chromosomes would be lead to segregation of a pair of factory or pair of factors they can carry. Means generally on the way, according to Boveri and Sutton, this tolls, they explains that the pairing of separation means pairing and separation of a pair of chromosomes, right, can always helpful for the segregation of the pair of factors they carry. मीन्स दोज उन क्रोमोसोम को पेयरिंग होना और उनका सेपरेट होना बिल्कुल क्रोमोसोमल थ्योरी के बेसिस पे ही होता है, ताकि क्या है जो पेयर ऑफ क्रोमोसोम्स हैं वो इस प्रोसेस को कराने में हेल्प कर सकते हैं, ताकि सेपरेट जो फैक्टर्स हैं जो उनके ऊपर कैरी करते हैं उनको आसानी से एक से दूसरे में ट्रांसफ़र किया जा सके. एज यू कैन जस्ट सीन इन द डायग्राम in the such structures, means you can seen over here that they are the homologous as well as the analogous one. After the meiosis first stage they can be separated out, means gamete formation are done over there, right, and after when they can be just form a single cell, right, they can be just separated and exchange their structures, means they, as you can just see the changes of the structures, means generally in such a structure or such a way. And these both can be done with the help of the different ways through which they can be separated out. For the such chromosomal theory, generally they have just performed, or we can say the experimental verifications can be done with the help of Thomas or T.H. Morgan, right? And this T.H. Morgan, the scientist, he done a experimental conditions with the help of a fly that is called the Drosophila, or means melanogaster. Means this is a fruit fly. Means T.H. Morgan had done his experimental phase on the Drosophila melanogaster. Generally, first of all, you all must know why he selected such a fruit fly. आखिर उन्होंने इसी फ्रूट फ्लाई को ही क्यों लिया, राइट, फॉर हिज एक्सपेरिमेंटल प्रूफ. जनरली ही सेलेक्टेड दिस ड्रोसोफिला melanogaster or fruit fly because they can, he explained that they could be grown or simply artificial medium in laboratory. Means to grow of this Drosophila melanogaster is easy into the laboratory or into the artificial medium, and the life cycle of the Drosophila melanogaster is very short, right, means they have at least 2 weeks life cycle. Only we can say the single mating could be produce a large number of flies, means whenever the mating can be done between these male and female Drosophila melanogaster, they can be capable to provide a large number of the offsprings in that particular conditions. So such it is easy to do the such process. Evenly we can say they can also show the clear cut difference between the male and female, because we can say the male are the smaller and the female are the bigger one. Means on the basis of their size we can easily identify them that male are the small size and the other, that is called the female, they are the bigger in size, right. So with the help of identification of them it is also very easy, as you can just see in the diagram, right. So with the help of this you can say that this is the female and this is the male. So by seeing them you can capable to easily identify that they are the male or they are the female. On the other hand we can say that their hereditary variation can be also easily seen through the low power microscope. Means generally their structures, their variations can be easily to recognize with the help of very low power of microscope. Means with the help of a low power microscope we can capable to identify them also. So as we are talking about the experimental of the T.H. Morgan, right, later we also explain about the another thing that is called linkage and recombinations, right? First of all, we have to understand about what is that linkage, right? Means generally, if we can talk about this, linkage is we can say it's a physical associations of the two or more genes on a chromosome. Means how the genes are attached to each other, right, on any chromosome, that is called linkage. And we can say that when two or more genes are closely located on a chromosome, that is called linkage. And we can say that those genes try to go together in next generations. Means generally the genes which are tightly located, tightly attached to each other, and they trying to live together, they will also try to go with in next generation, right? So that is, we can say, that inheritance is known as that type of inheritance of genes is known as linkage. I think everybody understand. You must know. But this condition is exceptions of the Mendel's law of independent assortment. It cannot be generally structured in such a way, right? Especially you must again say, generally when two or more genes are closely located on the chromosome and both genes try to go together in the next generations. जब किसी भी कोई दो या दो से ज़्यादा जीन जब एक ही क्रोमोसोम पे लोकेट करते हैं और वो सारे क्रोमोसोम एक्चुअली नेक्स्ट जनरेशन में जाने की कोशिश करते हैं, तो उस टाइप के इनहेरिटेंस को कहा जाता है क्या? लिंकेज कहा जाता है, राइट? जनरली वी कैन से इट कैन बी ऑल्सो means it can, the linkage is based on the frequency of the crossing over, means how the crossing over is occurs. If we can talk about the crossing over, generally the crossing over in prophase or meiosis first, where the homologous chromosomes break at identical to contain a region at each other, means they can be separated out from each other. And the linkage can be always done or realized or direct with the help of the test cross. We can say the linked genes are used for the mapping. They are found as the looking for the dimension from the frequencies, except from independent assortment, as I have already told you. Later on, generally these can be source, means generally no linkage is found in the independent assortment law, means we can say linkage can be occurs without recombination in that conditions, right? So let's move the linkage with the recombinations, means how the recombination linkage can be occurs simultaneously with each other, right? So for that you all must know that how they can be just transfer these characters from one generation to another generations, right friends? And you must also know the types of linkage, that is complete linkage, which is given by Morgan, T.H. Morgan. He explained the genes located on the same chromosome do not separate and are inherited together over to generation to right generations. And due to absence of crossing over, the complete linkage always allows to combination of the parental traits inherited as such. You can say during this complete linkage, the crossing over will not take place, the characters cannot be separated out from each other, so they can be directly transferred from one parental to their offspring also. And we can say it is a rare but has been reported in males, Drosophila and some other homologous organisms. And there is another condition is also found over there, that is called incomplete linkage. Means we can say incomplete linkage generally when the genes are present in the same chromosome have tendency to separate, means they cannot be transferred directly from one generation to another generation. Due to the crossing over, they can be just separate and they cannot produce recombination progeny beside the parental type. And we can say the number of recombinant individuals usually less than the number of expected independent assortment. As we can say in the independent assortment, all the four types or two parental type and two recombinant type, they can be formed at 25%, as well as in the linkage case the each two parental types is more than 25%, while the other recombinant type they are less than 25% in that condition. Then we have to talk about the genetic recombinations. We can say that recombination can be explained from generation of non-parental genes combination, right? Means we can say that genetic recombinations, it can be productions of the offspring with combination of traits that can be different from those found in the parent. Means we can say that when the genetic recombination, evenly the genes can be combined, but due to which the characters which are found into the parental but they are not found into their offsprings, so that is can found known as recombinations or genetic recombinations, right? You must prepare it that the genetic recombination, the production of offsprings, new organism can be developed with the combination of trait that differ from those found in either parents. Means जनरली जो पेरेंट्स में करेक्टर और जो जीन्स पाए जाते हैं उनके अलावा जो कॉम्बिनेशन बन करके उस ऑर्गेनिज्म के अंदर डेवलप कर रहे हैं, दैट इज कॉल्ड जेनेटिक रिकॉम्बिनेशन. Especially we can say, right, during in the eukaryotes, the genetic recombination during the meiosis can be lead to novel set of genetic information that can be passed on the from the parent to the offspring, and we can say the most recombination naturally occurring, right? In the modern genetic analysis, the main test for the determining whether two genes are linked based on the concept of the recombination. Means we can, in modern genetics we can find that whether the two genes are linked or not on the basis of the recombination they can be found. And if we can talk about the recombination frequency, then we can say the independent assortment always produces a recombinant frequency of 50%. For the genes to close together on the same chromosome pair, a physical linkage of the parental is recombination. So they can capable to make independent assortment impossible, and hence produce a recombinant frequency slightly and lower than 50% in that conditions. And if we can talk that on this frequency, it can be always under formula that is the number of recombinant and total number of progeny where they are found over there. So with the help of this we can capable to calculate such structures over. We can say that these means generally they can be always show the different conditions, right? So we can also find or known as that for the explanation of linkage and recombination, Morgan can done out several dihybrid cross in Drosophila, right? On the basis of his study, he can explain the different things, right? That the genes are located on the X chromosomes. Generally, due to that conditions, he can explain, he observed that two closely located genes did not segregate independently of each other. Means generally, जब कभी भी दो closely related genes किसी भी जगह पे locate होते हैं, तो they cannot be segregated independently. वो अपने आप आसानी से एक दूसरे से अलग नहीं हो पाते. First thing. Another thing we can say, the proportions of the parental genes combination were much higher than the non-parental. Means जो parental genes होते हैं उनका combination higher होता है और जो non-parental genes होते हैं उनका combination lower होता है, right? जो we can say whenever the two genes in the dihybrid cross were situated and on the same chromosomes. The Morgan concluded this by the physical conditions, right? And he joined with the linkage. Generally we can say the cross A shows the crossing between genes Y and W, as well as the cross B shows the crossing between the genes W and the M. Means as you can see in your diagrams, right? You can just understand with this one. And due to this, the dominant wild type of allele is represented with plus gene sign over there. You can just see in your diagrams in your linkage, and we can directly say a linkage is directly proportional to 1 by distance between the two genes. So due to these conditions, we can easily understand, we can easily say that when the genes are grouped on the same chromosomes, some genes were very tightly linked, so there is a very low recombination, while other which are loosely linked, they are very high recombination, right? As well as we can say the recombination of linked genes is by the crossing over, exchange of corresponding parts between the chromatids and the homologous chromosomes. So such a way they can show that structures. So student, you must see in your diagrams in cross A and cross B, where the male and females are over there, right? In the first condition, where generally they cross with the yellow white with the wild type, and wild type they are the dominant characters, and they can be show their structures, right? In the other cross B, where the again the male are dominant type, and they can be generally white and maniated and wild type one. In that condition, they will get the such structures. So students, next we can talk about the how that this can be helpful for the mapping one.